Okay, for those who do not know me, my name is Chelsea Maroff, and I teach a few of the labs for Bio 2 here at UNO. And I just wanted to make a video to help my own sections out. I'm used to being able to give you some guidance for the second lab report in lab after we have done the first one and you do your data collection for the second one. Obviously, we cannot be together to do that, so I decided to just make a video. So I sent out that survey to ask you, what you most need help with. And so hopefully this video will address most of those things or most of your questions that you have. So before you even start writing your report, I kind of laid out some steps that I think that you should go through just to kind of help you organize what you need to do to be able to write a good report. So the first thing that I would do is go and read Lab 9 in your manual. That will give you a really good feel for what you would have been doing in lab if you were actually in lab collecting this data. And then I would go and watch the videos on Canvas posted by Dr. Robbins. He made 2 separate videos. They're about 30 minutes total. And he uses a PowerPoint that is on Canvas as well. So if you go over to Canvas, you'll see all of the stuff that you need right here. Don't forget to take the quiz. And then this lecture is where Dr. Robbins uploaded those 2 videos, and he goes over like specific details on the organisms that we're using, how to do the dissections. So you should have a really good idea of how this experiment would have actually gone had you been in lab. And in this Lab 9 presentation slides is that PowerPoint that he uses to go through those steps with you. Your data that you need to actually use to write your report is right here. So that's class data. Everybody is using the exact same data for this report. It's just easier than us trying to figure out, how to use different data for each of the sections since you're not actually collecting anything. This making a bar graph with standard error bars is still there, so you can use that. I'll go over that a little bit in this video as well, if you just kind of need a visual of how to do that. And then when you go to actually upload your second lab report, please be sure to just attach this rubric to the end of it. Since we have to grade everything online, that makes it a lot easier for us to keep track of your point breakdown and to give you, a little bit of feedback, at least on your report when we hand back that. So this is Dr. Robbins' presentation. He goes through a lot of information on this lab, and he has examples of graphs and what those captions should look like. So I don't want to spend too much time on these details today because he has those already prepared for you. Some of you did mention that you could use help on those things, so just watch those videos that Dr. Robbins has made. He already outlines how to do that. I'll just briefly cover a couple of those points here, so you just kind of have all of these details in one place. After you watch all the videos and you've read the manual, then I would go and actually just read the actual instructions for how to write your report on pages 203 and 204. I think that'll be very helpful for you to kind of get organized on how you're going to do things. Then start your report, after you've already done all the research, as if you had been in lab, basically is kind of what we're trying to mimic. Then you can come back and watch this video if you still need some added support, and then use that rubric. It's in your manual, and I also posted it on Canvas there under the assignment, so you should be able to find that. Use that as a checklist. When we grade these reports, I know at least for myself, I'm not really grading expecting you to be an excellent scientific writer. I know you don't have a lot of experience with scientific writing, so I'm not expecting you to have a beautifully written report. I'm more just grading for content. So please, please, please use that rubric to make sure that you have all the details that you are supposed to include in your report. And then make any final edits. At any point in this process, you're welcome to email us, ask us questions, set up a Zoom conference, whatever you need to do. I don't want you to have added anxiety just because you can't be on campus. So, we're here to support you, and anytime this week, send us questions that you have. If you want to start a Zoom conference, we can do that as well. But in this PowerPoint, I'm going to go through the things that were basically most asked for with like what you need help on for this report. And so I'm going to briefly cover what you should include in each section, not specific details for this report, just kind of in general how to write a report. I'll go over how to make your graphs, how to decipher your graph, and then commonly missed points on lab report 1, which you can hopefully avoid for lab report 2 and get all of those points the second time around. So for each of these sections, I'm going to put links down below so you can just jump to the sections that you need help on. If you don't need help on all these sections, if you already know how to make the graphs, that way you can just skip those sections in this video and not spend too much time here if you don't need to. And that way also, if you've watched this video already and you just want to go back to reference things later, those links will be down there that you can just jump to what exactly you need to go through. So for what to include in the introduction, this is pretty well laid out in the rubric and in the instructions in your manual. Obviously, you just want that background and purpose section. And so in the introduction section, what I say, and kind of the general rule, is that you want to start broad and then get more specific. So it should look kind of like an inverted triangle, essentially. And so think of that in terms of you want to start as broad as possible and then basically get to your hypotheses at the end of that introduction. Many of you did that. There was just kind of a disconnect between your sections. And so what I would suggest, the main thing here that you might want to try is to weave your hypotheses into the actual introduction paragraph that you write. I think this will help with organization. It'll help you link your background material to your hypotheses and kind of avoid some of those common mistakes that people make. One last thing here, be sure that you cite your manual when you are using your manual for information. So I wrote on a lot of reports this time around, source ? In your introduction paragraph, and that's just because you used material obviously that came from your manual or lecture and you gave no citation for it. That's plagiarism, and that's something that we really want to try to avoid in scientific writing. So if it's not common knowledge, then you probably need to cite it. So obviously, for this experiment, fish live in water and rats live on land. So that's not something that you would need to cite. But if you're talking about specific details, like if they're ectotherms versus endotherms and what their diet is and how their environment affects that, that is material that you're going to need to cite. You can use your manual, that's perfectly fine, but just don't leave a bunch of information in your introduction paragraph uncited or unaccounted for. And then the big thing that I try to push in the second report, since you've already had one go at this, is trying to use your background material to support your hypotheses. This is really the whole point of the introductory paragraph. You kind of want to lay out some groundwork for, why you're studying this in the first place. So why are you looking at these 3 species, frogs, fish, and rats? And then use the background material that you present to support your alternative hypotheses. Your hypothesis should not be unfounded on any evidence, right? You want to use material that you've already found in other articles or your textbook or your manual to back up what you predict to happen in your experiment. Okay, for the method section, ask yourself, would my instructor be able to duplicate my experiment after reading my method section? If the answer is yes, then you probably have a good method section. If the answer is no, you're probably missing some details. So think about key words here, like how you did the dissections and what basically units you used to measure those things. So did you measure with a ruler or measure with a scale? Like, was it weight or length? You should be able to get all that just by reading through the manual. It's pretty straightforward. Include and explain some keywords like snout to vent length. Those things that are like new terms for you, maybe explain them a little bit, like briefly in one sentence what they mean, and then include things like species names. The last detail here that you might want to include, definitely need to include, I should say, is sample sizes. And I'll talk about this a little bit later, but don't say 5 groups looked at rats and 5 groups looked at fish. Pretend like you're the one that collected all this data, or just pretend like your lab section was a collective research group. And so you can just use the word we, like we did this. And so we looked at 5 rats and measured the heart, stomach, and intestine in them. That's your sample size. Your sample size is 5 rats, not 5 groups, because I don't know what you mean when you say group. And so I had a lot of people who used the word groups for their stomata data. And so I maybe made a note on your paper like, What are you talking about? Did each group have their own plant, or how did you do that? So just be specific there so we know what your sample size is. For your results section, please do not put raw data here. That's one big thing that I saw in the lab report for the first one. That's the point of your graph, is basically displaying those numbers. You know, humans, we're not that great at just looking at raw data and being able to visualize in our mind what those trends actually, you know, come out to mean. And so that's the point of our graph. So putting the raw data in your caption or in your results section isn't really beneficial for everybody, for anybody. And so you can just leave it out. That's what your table is for, your graph. But you definitely do need to explain what your standard error bars mean. So talking about whether or not they overlap and what that means, and then how that relates to your hypotheses. And so a lot of you said that, you know, you've rejected your null hypotheses. Most of us rejected all of our null hypotheses for the first lab report. That's pretty common. Probably the same sort of thing for the second report. You know, we expect to see differences. That's why we're doing the experiment, right? But you didn't follow up on that and say whether or not you supported or rejected your alternative hypotheses. And that's really the big question, guys, for these reports. I mean, you're making hypotheses, and that's the basis for your entire report, right? So you definitely need to follow up and say whether or not you rejected or supported your alternatives. It's possible that you are going to reject your nulls and not 100% support your alternative hypotheses. It's possible that you're somewhere in between. So there's a little bit of gray area on that. Just say whether or not you completely supported your alternatives or not. And you can reject your null and also reject your alternative, but just make sure you mention that in your results section. When you get in the discussion section then, this is where it gets difficult. And I don't grade super harshly here because I know that this is one of the tougher sections, this in the introduction. So this should be similar to your intro, but in the intro, you know, we started broad and got specific. You're gonna want to invert that triangle for the discussion section. So you're gonna want to start specific and then get more broad as you move throughout that section. So these three key things that I would discuss in this section I have right here. What were your results? So kind of interpret your results, say what those results mean for those species, and then how do those results relate to the real world? Be very, very careful here. You can't explicitly say, you know, we put a plant, a spiderwort plant, in a dark box. That means photosynthesis stopped, and that's why the stomata closed. I saw a lot of things like that in the first lab report. That is not actually true, or you can't really say that. You can say that you think that that's the reason, or that's like a plausible cause for the stomata closing, but you didn't really measure photosynthesis, right? So you have to only talk about, you know, the things that you can explicitly support or not with your results. And then trying to relate those, you know, to the real world. So just basically in that term, you know, kind of think back to the species in their environments, you know, kind of how you talked about things in the introduction section. So like diet and environment and evolutionary history, and how your results support or maybe kind of go against what you would normally think for those factors. Okay, I'm gonna go through a little bit of the graphs now for the how-to, like how to build a graph. So if you already know how to make a graph and you just want some help deciphering it, just skip to the next section. But I want to show people how to do this because it's much easier for me to grade if you have these correctly made in Excel. So I'll give you a few tips here. So for this report, you're gonna need 3 graphs. You should have just had 1 for the first report. And I'll show you how to make the stomata data one. And it's going to be very, very similar for your second report. So you're just gonna have 3 now instead of 1. And then there's still that Word document on Canvas that you can use for making the standard error bars. I'll go through it briefly here just to kind of give you a visual on how to do that. So we have here our stomata data. And so your section won't have, you know, exactly the same data as this, but it probably was very similar. I think this was Wednesday AM data. So you can see I've already pulled out, you know, my treatments, and we did this for you in this Excel spreadsheet. And then I have the means that we pulled out from the effect on stomatal opening. In other words, our change in stomatal opening is what we wanted to use for that one. So we pulled those out over here. We pulled out the standard errors for each of those as well. So if I just highlight simply these two columns. That's all you need to do. Highlight your columns, go up to insert, and you can just do insert a 2D column graph. And you can see that's basically a completed graph already. So it's not that hard, but maybe you're not that comfortable with Excel. So I just wanted to give you a little bit of help on that if you need it. We want to change a few things, obviously. First, delete this title. We don't use titles in scientific papers, so just delete that right off the bat. Since this is negative data here, I can, if I want to, tell it to put the labels at the bottom, but for this second report, you shouldn't have any negative values, so that shouldn't be an issue for you the second time around. And then we can go in, and you definitely want to add some axes titles, horizontal and vertical. Obviously you have units up here, and then you have your treatments down here, but those are not axes titles. You just want to add those as well. So we would call this treatment, and this would be average to model change. You have to put the units in there. So if it's like millimeters or whatever you're measuring for your animal experiment, but we wanted to put the percentage on there. That's our unit for this graph. And then what's the big thing that I'm missing here on this graph? The standard error bars, right? So if you highlight your bars, that's going to highlight your data. Then you can tell it to add error bars. But if you just have Excel use its standard error bars, they're not going to be correct. And that's basically because it's just making standard errors based on this whole data set together. We wanted to have individual standard error bars for each treatment. So you still go up here to add chart element, but then you want to click on the more error bar options. And you'll see way down at the bottom here, custom error bars, and it automatically sets them all at 1, so you have to specify the values. Click on this button, it'll allow you to highlight data on your spreadsheet. And we've already put our standard error bars over here, so we can just simply highlight all 4 of these numbers. They're in the same order that our treatments are in, so they're already going to be correctly applied to each of these bars. Hit enter, do the same thing for the negatives. You don't need to actually type in a negative sign. Excel knows that you're using the same values and they just need to be automatically negative. Hit OK, and look at that, beautiful standard error bars correctly displayed on our graph. You can, edit the color. I don't really care if you change some of the details on here. You can mess around with this a little bit if you want to make your graph a little bit prettier or a little bit different. But that's basically the gist of what you need to do. And then you can actually copy. So if you click on the outside border here, you can copy, hit Command C on a Mac, or you can come up to the top here, copy that, and you can actually, I'm going to go to a Word document here. You can actually paste that directly into a Word document. So a lot of you uploaded pictures. I think this is actually much easier. You don't have to hand draw a graph. And then if you click on your graph, and then you can actually, I'm going to move this down here. Okay, so if you go to chart design, or format, I should say, you can tell it to arrange in a different manner that basically allows you to move your graph around. Obviously, you want to be able to move your graph into where you want it on your paper, and you can basically perfectly center it over your figure caption then. One last thing that I would say about this section, for your results, you want to put the paragraph of text for your results first, and then put your graph second. It's a lot easier for me to pick apart like where your sections are. Otherwise it all kind of start to run together. Looks a little funny. It's hard for me to keep track. So put your paragraph first, put your figure actually at the end of your results section. I know that seems kind of counterintuitive, but that's pretty standard. And then your figure caption, please do not forget your figure caption. That's worth a good chunk of points. And you want it to be pretty, detailed, but not more detailed than needed. So again, don't put any raw data here. Basically, I just want to tell my readers what are these bars that they're looking at? You know, what were these treatments, and like what was I actually measuring, and what are these standard error bars? Just kind of go over each aspect of this graph. And so I said down here, responsive spiderwort, stomata in a control environment, and 3 different environmental treatments after 1 hour. So I'm telling them how long I ran my treatments, what species I was looking at, basically using in my experiment, and then what was I measuring, and all that's just in 1 sentence. So this doesn't have to get super long, but you do want to include all those details. And then I just said, you know, what my bars are, so they represent average change in stomatal opening as a percentage. And then the treatments include high humidity, warmth, and darkness. You can include a little bit more on the treatments if you want to, but you don't need too much detail there for this. And then just tack on to the end, standard error bars represent plus or minus 1 standard error. That's what we use for this lab. It's not that common, but that's just an easy way for us to just give you a statistic to get used to. So that's all you need for your graph in your paper. So if you have any other questions on that, just let me know. Just remember to take off the title, add the axes titles, make sure your standard error bars are correct, and then have a good caption. Dr. Robbins has an example of a caption in that PowerPoint. You can look at this one in my PowerPoint. There's another one in the manual from your first actual lab exercise. So you have a lot of options that you can go to look for details on what you need to include there. All right, so when you actually go to decipher your graphs, I know that this is a little bit tricky for some people, but I think that most of you have a pretty good handle on it now. I just want to go over it one more time just to make sure that you are completely solid on understanding how that works. And so to decipher your graph, we're basing it all on the standard error bars, right? So we have the average stomatal opening percentage over here. We have our treatments down at the bottom. And so if I have a mean, like humidity is right here and darkness is over here, you can see that these standard error bars clearly overlap. So what that means is if I was to do this experiment again, the same exact experiment, I might expect to get a mean for humidity anywhere within this range, because I had quite a bit of error between all of the leaves that I was measuring. So that standard error means that you can have a mean basically anywhere in here. Same thing for darkness. In other words, these means, the next time you run that experiment, could be exactly the same. So you can't say that they are statistically different. So there's no significant difference between this treatment and this treatment. However, if we compared humidity to warmth, clearly there's no overlap in those standard error bars. You could say that those are significantly different from each other. So the whole point of this is whether or not you have significant difference between your treatments. And for the stomata data, we wanted to compare it to the control. For this animal report that you're doing, you're going to have to compare it to basically each other, the species to each other. You don't really have an actual control. That's okay. The concept is exactly the same on how you do that. And so, you know, when you're looking at your animal data, you should have very, very similar graphs. You won't have any negatives, so that'll kind of help get rid of that issue that some people had there. But you're also going to have 3 different graphs. And so for graph 1, you're going to want to compare the heart mass, the relative heart mass for fish, frog, and rat. And so you're comparing all 3 of those species to each other. So it's not comparing just, you know, experimental treatments to the control. We're comparing, you know, 3 different groups to each other. And so it gets a little bit more tricky. It's more likely that you're not going to fully support your alternative hypothesis if your alternative hypothesis is very specific, but that's okay. You know, sometimes experiments are just a little bit more complex, and that's just kind of the way things work. So if you need any more help there, just let us know. But just remember, you're just looking for overlap in your standard error bars. So you want to be sure that you have a correct graph before you start this section. So if you're still unsure on the graph, let us know sooner than later so we can point you in the right direction. Okay, so finally, I'm just going to go over a few common mistakes. You can refer back to these bullet points and hopefully, you know, avoid making those mistakes for this time. So make sure you have specific hypotheses. So a lot of you said your null was that there was going to be no difference, which is great, that's fine. But then you said your alternative hypothesis was simply that there was going to be a difference. I want to know where that difference is going to be. That's kind of the whole point of this report, is that you're kind of predicting what you're expecting to see. So make sure your hypotheses are specific, your alternative hypotheses. So don't just say, I think that heart mass is going to be different between fish, frogs, and rat. Say something like, I think that fish are going to have the biggest heart mass compared to the other two, or you can even rank them. Say that I think fish will be biggest, followed by frog, and then rat will be the smallest. So kind of, you can decide how you want to go with that and how specific you want to be. Like I mentioned when I was talking about what should be involved in each section, make sure that you cite anything that is not common knowledge. Cite your manual wherever you need to. That's perfectly fine. When you go to cite your manual, just look at page 7 of your manual. So I just scanned this into my computer. So Dr. Robbins actually has typed out for you exactly how to cite your manual. So you should be able to do that. You'll just need to change this first part because you're going to be looking at lab 9 or chapter 9 instead of chapter 1, which I think this was from. The rest of this will, for the most part, stay the same, other than the page numbers, obviously, here. And I think that we're on the 2nd edition of this manual now. Let's see. Connect your hypotheses into your background info. I think that that'll kind of be the best way to do that, is to just make sure you have your hypotheses within the text of your introduction. Don't just list them as bullet points or numbers below your introduction. Method section, just ask yourself, would my instructor be able to replicate my experiment? And in sample sizes, just don't use the word groups. Just say how many specimens you were looking at. Results include null and alternatives. So did you support or reject your alternatives? That's kind of the most important thing. You want to say whether or not you rejected your null, obviously, but then you need to include that alternative hypotheses in there as well. And then I think that there was just a little bit of disconnect on the standard error part. I had a few people say they rejected the null and they supported the alternative, but then they still said that the error bars don't overlap, and that means that they're not different. I mean, the key here is that if they don't overlap, that means they are significantly different from each other. We're really just looking for differences in this data. And then just don't put raw data there, and that should be good. For the discussion, if you missed points there, you probably missed points because you didn't talk in fully about each of the experimental treatments you did for the stomata. So if you did this whole experiment and then at the end of it, you only talk about temperature in your discussion, that just is not a complete paper. You need to make sure you tie everything back in the same way you do in the introduction. Talk about each species and each organ in all those organisms that you're looking at. And then a couple more mistakes that I saw, just use past tense throughout, even in your method section. Just say, we did this, we did that. You don't need to use you can just pretend like your whole class was like your research team. And then for the citations, I'll just show you how to do this really quick. I don't want to see any more mistakes on notations because on the citations, just because it's so easy. So I just want to make sure you know how to do that. So if you go to page 204 of your manual, we again gave you a list of articles that you can use for this report. So you don't have to find your own if you don't want to. So go to Google Scholar. So I just typed in scholar.google.com, and then I'm just going to type in that first reference. So Piper J, 1982, respiratory gas exchange. And you don't even need to type in whole things. Lungs, gills, tissue. That should be enough. There's my article. I can just click on this link right here to download it. And then this quotation, this magic quotation button down here, gives you the exact citations that you need. So please do not make any more mistakes on this, because all you need to do is go to Google Scholar and just copy and paste. Sometimes the formatting gets messed up. So if you notice things are in all caps or everything's italicized, that's an easy fix on your end. Just make sure that that's correct when you go to copy and paste it. So you can hit Command C, go up to the top there, and then you can go back to your Word document. This section should probably be called literature cited. That's the standard. And then I like to just say match formatting when I paste so that all the fonts stay the same. And then just tab this over, and there is your citation. So hopefully you won't have any more issues with that. Now I'm going to guess that this is probably many of you right at the moment. So if this is you, please don't wait until the last minute to write this report. We want to help you as much as we can. I know that it might be stressful for you because you don't have that in-person contact and you're not actually running the experiment yourself, and that's tricky. But it's a good experience, I think, for all of us to be dealing with this right now. Just make sure you start this sooner than later. If you wait until Sunday night and you're trying to email your instructor to ask questions, if we have like 50 emails that we need to respond to, we're not going to be able to get to everybody and answer all of those questions. So asking us sooner than later is better for you and us. So if you have questions, email me if you want to. Even if I'm not your instructor, if you have questions over this video, feel free to email me. For my sections, I'm probably going to set up Zoom sessions on Wednesday and Friday that you can just sign on, ask me a question quick if you want to. If you'd rather just do it over email, that's fine as well. So just let us know, and good luck with your second report.